Hello, thank you for joining me today. My name is Debra Buggs, and I'm a sophomore biology major at Oglethorpe University. My topic focuses on Burmese pythons, how and why they are a problem, and also what steps are being taken to manage them. Today, I will be discussing the use of ADLs to study fine-scale activity of invasive Burmese pythons in the Everglades. I became interested in the topic of invasive species last summer. During quarantine, I spent a lot of time going on walks and viewing natural wildlife. One thing I noticed is a lot of plant life that I didn't recognize. After doing a little research, I found that they were invasive plant species. So when choosing my topic, I knew that invasive plants was the way I wanted to go. When narrowing down my topic to be more specific, I knew I wanted to keep it in Florida since that's where I'm from. Turns out it was very hard to find papers on just one type of plant. So instead, I did a Google search for the most invasive species in Florida, and I came across the Burmese python. Being from Florida, I found it really interesting that I was not more aware of this problem. So even if you're not a Florida native, I hope this presentation will give you some insight to invasive species and maybe get you more motivated to learn about the ones affecting your area like it did for me. Here's an outline of what we're going to be talking about today. We're going to get started with some background information so we can fully understand the impact of invasive species. Then we will talk about a study that utilizes a new type of tool to study pythons. We'll go over their introduction, some methods, their data, the conclusions that derive from the data, and finally, we will end with some take-home points. Let's get started with our background. Here, I have a list of terms I want you to think about as we go through the background slides. Before we move on to the study, you should be well-versed in what invasive species are, why they are a problem, and finally, what methods are being done currently. We will also discuss Burmese pythons, the Everglades, and range expansion potential, which is an important factor in managing invasive species. First on our list of terms was invasive species. Invasive species is defined as any living organism that is not native to an ecosystem. One example of an invasive species are invasive snakes, which are a growing problem in tropical to subtropical climates worldwide. This is because they prey on a lot of species and encounter very few predators. Let's take a look at the figure to the right. In the middle of the figure, we see an invasive snake. When we introduce this snake to a non-native territory, there are very few animals that will prey on it. The arrows point to animals that the invasive snake may prey on. Here we have bird species and small mammals. The double-headed arrows indicate mutual predation. So looking at this simplified example, we can see that snakes will eat or harm 4 other species, but only 2 will hunt and kill them. Now imagine this on a much larger scale, with the number of prey increasing, but the number of predators remaining at only 2. This directly correlates to the enemy release hypothesis, which states that since the snake is introduced to a non-native territory without its natural predators or natural enemies, they're able to become very abundant. We can see why invasive species are such a problem. In the case of invasive predator snakes, another reason they are really problematic is because they are very difficult to detect. Let's sum up what we have learned in this slide. Invasive species are any organism that is not native to an area. One subset of this are invasive snakes, which pose a problem worldwide due to their wide variety of prey sources, lack of predators, and their slickness when it comes to detection. In the previous slide, we talked about invasive snakes and learned about why they are such a problem. Now, let's talk specifically about the case of the Burmese pythons in the Everglades. Before we look at these figures, let's talk about Burmese pythons. They are predatory snakes that can reach up to 6 meters in length. They can live for a very long time, from 15 to 25 years, and have a wide range of things in their diet. Another thing to note is that they produce very large egg clutches, ranging from 8 to 107 eggs per clutch. So, where do they come from? Let's look at this figure on the left. This is showing us the origin of Burmese pythons. This is a map of Southeast Asia, and the green parts are highlighting places where Burmese pythons are native. Burmese pythons arrived in South Florida through pet trade, imported initially from Bangkok, Thailand, the red dot under Thailand, and also Ho Minh Chi City, Vietnam, which is here. They have most likely been breeding in the Everglades for about 30 years. Their invasion can mostly be attributed to pet releases and also Hurricane Andrew in 1992. The Category 5 hurricane destroyed pet breeding facilities and released a large amount of pythons into the wild. The Florida python has shown very little genetic variability and appears distinct from the Vietnam population. The scientists have predicted that the Florida population has mostly Thai origins, although more DNA sequencing would be needed to determine where exactly the Florida population originated from. So what is the current range of pythons in the Everglades? The Everglades is actually the largest subtropical wilderness in the United States. It is home to a numerous amount of wildlife, including various endangered species. This includes the manatee, American crocodile, and the Florida panther. So let's look at this figure on the right. This is showing us a map of South Florida. The green area is showing us the Everglades National Park. Purple triangles represent presence points, meaning places where pythons have been spotted, and the black dots are showing us some major cities. Here I have a red arrow pointing to Miami. This was the main spot where pythons were introduced to and released from during pet trade. One interesting trend on this map is this line of purple presence points here. If you look at a map of the Everglades, you'll see that there's a major road that goes through here. So the more people that are traveling on this road, the higher chance there are of presence of pythons being sighted, which is why we see this trend. I thought that was very interesting. Within the past decade, pythons have expanded out of the national park and into areas of higher density, which we can see that the pythons are starting to fan out of this green area, reaching as high as Okeechobee, even further north. This is important because when the pythons expand their population northward, this can cause problems for other environments, not just the Everglades. For the past 30 years, scientists have also seen a significant decline in mammal populations and also diversity in the Everglades. This has caused massive changes in the ecosystem, and thus the pythons must be dealt with. The problem is that pythons are hard to detect, and so little is known about their fundamental and physiological attributes. So how do we go about managing this problem? The answer to this is finding out their range expansion potential. What is range expansion potential? This is an important consideration for management actions against invasive species. Like its name implies, range expansion refers to the area in which invasive species can establish themselves, and potential refers to the possible places where the species can move to. Expansion depends on a multitude of factors. For many reptiles, their ability to thrive in an environment can mostly be explained by what temperatures they can tolerate. The lowest temperature the species can withstand is an important factor in determining range expansion potential. Another important factor is behavioral flexibility. If scientists can understand how the pythons will react to colder temperatures and what temperatures they can withstand, they will better be able to determine the range outside of South Florida to which the species can expand its population. Cooler temperatures may pose a threat to Burmese pythons because in their native homeland, Southeast Asia, the temperatures are generally stable and support their normal activity throughout the year. So what would scientists do with this information? Predicting range expansion potential can eliminate the breadth and magnitude of Burmese python's impact beyond South Florida. That leads us to the end of our background discussion. Just as a reminder, here are the key terms from the first background slide that you should know. This includes invasive species, Burmese pythons, the Everglades, and range expansion potential. Next, we're going to look at a study that uses a new type of technology to study Burmese python behavior so that they can get a better model of range expansion potential. The scientists in this paper use a new and improved method of wildlife tagging called accelerometry. Before we get into their methods, we're going to look at the title in depth and give a brief introduction to this new method. So when we look at this title, there are two key terms that we want to identify before we can understand the purpose of this study. The first is accelerometry, a technique that utilizes the device to quantify movement patterns. We can think of it as a more advanced GPS tracker that quantifies movements instead of just location. Now, we also have the term fine scale to describe the activity that the scientists will be monitoring. When the author uses fine scale here, they mean greater detail. The scientists want to know more than just the location of the pythons. They want to know their movement patterns and other habits that only an accelerometer can detect. Now that we understand the title a little more, let's take an in-depth look at the rationale behind this study. Looking at the figure to the right, on the top, you can see the actions that have been taken in the Everglades. This includes detection dogs, traps, visual surveys, and GPS tracking. One GPS tracking method that has been utilized a lot is Judas Python tracking, in which snakes have a tracker attached to them to reveal the location of other snakes, which is most useful during breeding season. However, the primary tool so far has been human visual searching. However, these actions have proved not very useful. It's been 30 years and we still don't know much about the pythons. The problems that scientists have when they're trying to use these methods is that they don't know their behavior patterns. For example, they don't know when snakes are most active, during the day or during the night. It's easier to detect the snake on the move. We also don't know their predation frequency. We have only inferred from the decrease in native populations what their prey species are. And it's hard to trap a python when you don't know what exactly to bait it with. The cryptic nature of pythons also makes them difficult to study. They are very well camouflaged. And on the second row in the picture on the furthest right, there is a Burmese python and some foliage. I know when I first looked at this picture, it was very hard for me to actually see where the python was. So it's easy to understand why scientists are having a hard time locating and studying these animals. Radio and GPS telemetry have also been used in the past, but many snake body movements and behaviors do not produce a change in signal locations. And also, due to battery constraints, they only provide locations on scale of hours. Pythons are also known to select habitat with thick vegetation, which can often deflect the signal from transmitters. Therefore, scientists have concluded that there is a strong need for fine-scale monitoring of activity and behavior of the Burmese pythons. And the proposed solution is ADLs, which stands for Acceleration Data Loggers. These have been used as wildlife tags for other animals, but not large-bodied snakes. So to establish a technique, the goal of this study was to establish a technique for monitoring the activity of large-bodied snakes in the wild, using the Burmese python as the model, while also attempting to identify behavioral patterns that could be used to help manage invasive species in the Everglades. The question they wanted to answer is using ADLs an effective way to study large-bodied snakes? They hypothesized that the ADLs will be an efficient method to obtain information about behavior in the snakes, and they expect to see measurements for fine-scale body movements. Before we move on to the next slide to discuss the methods, I would like to give a little heads up for those of us who are squeamish. The ADLs used in this study are internally implanted into the snakes. Just know that scientists have no other option but to implant the ADLs internally. Because of the way the snake moves, an external harness would not be useful. But don't worry, these were done with the utmost care and under the University of Wildlife protocols. This was an observational study, so they didn't really have control or experimental variables to make comparisons. The purpose was to observe snakes using an ADL attached to them and see if we can get data on the small details of python behavior. So how do they go about doing this? Pythons were visually spotted and captured crossing roads at night or on the roadside during daytime searches in the Everglades National Park. Capture was done under University of Wildlife protocols. Now let's talk about the tag attachments and field trials. The ADLs used were the Vimco XYZ and CFAS G6A models. The CFAS G6A model is able to tag additional information, including pressure and temperature data. For animals with the Vimco model, an eye-button temperature logger was utilized. All ADLs were set to measure triaxial, meaning all three axes, X, Y, and Z, acceleration at 1 Hertz every 30 minutes during the 2011 field trials and every 30 seconds during the 2012 field trials. The reason for this was to minimize memory usage and maximize recording duration. This allowed the tags to record for approximately 3 months before their memory would be full. To ensure that they can maintain control over the ADLs during the entire duration of the study, the ADLs were surgically implanted within the lateral body wall, 2/3 distance from head to tail, to avoid puncturing the lungs. If we look at panel A in our figure on the right, the red and orange dotted line is showing us the placement 2/3 down the body where the ADLs were surgically implanted. And panel B is showing us where inside of the actual body that the ADL was placed. So to ensure the data logged by the ADL was actually a correct representation of the snake's movements, the loggers were held in place by suture material passed through the skin of the snake and through a single barrel aluminum crimp sleeve that was epoxy to the logger. If they had just let the ADL flow in the body, then it could move, and that movement would be recorded as if it were the snake, when it wasn't. It was just the ADL moving. So we can see how this looks in panel C, which is actually also showing us the relative size of the ADLs with their aluminum crimp sleeve and the suture material. And it's also showing that it's only about 4 centimeters. So it was very small. Once the snakes were released, their movements were observed and noted for several minutes so that the acceleration signatures from the ADLs could be verified. This is to make sure that the ADLs were working properly. They did the same thing when the snakes were recaptured at the end of the trials to make sure that the ADLs were working the same way. 2 VHF transmitters were put into each snake to make sure the scientists would be able to relocate and capture snakes at the end of the trial. Some snakes also had GPS data loggers implanted into them, and our locations were tracked every 1 to 2 hours. After 20 to 95 days, the snakes were relocated via their VHF transmitters, captured, and brought back to the lab for surgical removal of their tags. Now let's discuss the data. This is a raw 3-axis acceleration from one of the tagged Burmese pythons. We can see that the y-axis is labeled acceleration and that it's measured in Gs. It ranges from -1 to 1G. To put this in perspective, 1G is 9.8 meters per second squared, which is the acceleration of freefall. The negative and positive values here are indicative of direction. So in this case, positive values means to the right and upwards direction, and negative values mean to the left and downwards. The x-axis is showing the time and date of data collection. Data collection started at 1746 and lasted until 1754 on the same day. For those of us not familiar with military time, that's me, that is from 5:46 to 5:54 p.m. In the methods section, we discuss how the ADLs would be able to monitor the acceleration of snakes in all 3 axes. This includes X, Y, and Z. The purpose of this graph is to prove that ADLs did indeed do what we thought they would. The black line is showing us the x-axis, the red line is showing us the y-axis, and the blue line is showing the z-axis. One more thing to note from this graph is the blue sections, which highlight periods of non-movement, and the white sections, which highlight periods of movement. Now that we know what we're looking at, what does this mean? The scientists wanted to evaluate physical movement of the ADL versus behavioral movement of the snake. If you think about how the snake moves, it's moving like one section of its body at a time. So if you remember in the methods, the ADL was placed 2/3 away down the snake body. So when the snake is moving, even if it's one continuous movement of travel, the ADL might not be moving that entire time. It might not be recording acceleration. So to account for this, the scientists applied a filter to the data to show physical movement of the ADL versus behavioral movement of the snake. The tags also recorded their angle inside the snake. So if the tag moved less than 2.5 degrees for at least 7 seconds, it was considered non-movement. Any other motion that lasted greater than 3 seconds was considered as movement. This is where we get the blue and the white sections in our graph here. This is great, but they also needed a way to quantify brief, discrete movements, such as body repositioning and posture changes. Snakes were considered to be traveling to another location if their movement lasted longer than 20 minutes. Any other movement was considered as small-scale movements. These are changes in the body position or posture. We go back to our title. This is the fine-scale activity that they were talking about. Here is some more data quantifying the movement of pythons, but from a seasonal perspective. In the upper left corner, we have an abacus plot showing the activity of 4 Burmese pythons. On the y-axis, we have the different labels assigned to each snake, and the x-axis is showing us the month of the year. Each day is color-coded by the number of hours spent moving. The warmer colors means more movement, and the cooler colors means less movement. November through April, represented by the gray shaded background, is the presumed breeding season of the pythons. Some snakes were tagged different years, some in 2011, some in 2012, but they were placed onto one calendar year to show seasonality. The dark gray bar for P42 is showing us its second deployment, from which it died for unknown reasons. Now let's look at the plot beneath it to the left. This figure is showing transiting bouts for snake P42. By transiting, they mean what they deemed as traveling. If you remember from the last slide, we discussed that traveling was considered any movement greater than 20 minutes. So from now on, I'll just refer to it as traveling. On the y-axis, we have its displacement distance from the GPS, loggers, surge acceleration, and dominant traveling periods. And on the x-axis, it's showing us a time scale throughout the day that the data was recorded. Movement bouts often took place during nighttime hours, which is shown with the gray shading, and with dominant traveling periods of 3 to 8 seconds. So if you look here, we can see we have this yellow warm color in our dominant periods panel, ranging from 3 to 8 seconds. So when the snake was traveling, it did it in increments of 3 to 8 seconds for the most part. Now let's look at the plot to the right. This is showing us the percent of time spent traveling according to the time of day, and also the environmental temperature for 4 tagged Burmese pythons. Percent time traveling is represented by color across hour of the day on the y-axis and environmental temperature on the x-axis. All colors in this figure correspond to the percent time transiting legend in the upper right-hand corner. The top bar plot represents the percent time traveling across the environmental temperature range, and the right bar plot represents percent time traveling across hour of the day. At colder temperatures, less than 14 degrees Celsius, snakes displayed little movement behavior, but movement became more likely during the mid-morning when temperatures reached 14 to 20 degrees Celsius. Once daytime temperatures exceeded 24 degrees Celsius, movements became much less common. However, as temperatures increased even further, there was an increase in activity during nighttime hours. One of the goals of this study was to evaluate behavior of pythons. The different temperatures produced different behaviors in snakes. When it was colder, the snakes preferred moving during the day, but when it was warmer, the snakes preferred moving at night. Referring back to the abacus plot, snakes generally showed less than 2 hours of movement, and most movement was seen in the middle of the presumed breeding season here. Based on these data, the scientists concluded that snakes' behavior is heavily influenced by the seasons and times of day. Now let's discuss the conclusions. Fine-scale activity and some behaviors were apparent from the ADL data, meaning that ADLs are a potentially valuable tool for monitoring large-bodied snakes in the wild. Individual pythons spend 86.1% of their time resting, but often move continuously for several hours at a time during bouts of traveling. Individuals may shift their activity pattern based on the season, whether it's breeding season, whether it's warmer, or whether it's colder. Understanding these seasonal differences and activity levels can improve the accuracy of population estimates, help detect range expansion potential, and improve managers' ability to capture and find individuals. This data supports the hypothesis that ADLs would prove as a useful tool to not only track python behavior, but also help scientists and management officials with broader projects such as preventing expansion. This data and the conclusions they were able to derive from it may help promote management urgency. The more we know about their range expansion potential, the more they can push management ideas. Imagine if you lived in Central Florida and you owned a business in the farming industry and you own livestock. You'd be in more favor of management control on pythons if pythons ended up in your backyard. But if you didn't know it was a problem or that it could affect you, you wouldn't be in any rush to promote it. That's why this research is so important. That concludes our discussion of ADLs as a method to observe large-bodied snakes in the wild. Now let's finish with take-home message. Like me, you may not have been familiar with Burmese pythons or even invasive species in your area. However, I hope this presentation has taught you a little more about the importance of this topic. Before we end, I would like to leave you with some take-home points. In developing invasive species management priorities, it is not only important to gain insight into the potential ecological impacts, but also to assess the species' potential for range expansion. This is why the research we talked about today, as well as other methods of investigating invasive species, such as modeling, are so important. The more we know about the Burmese python, the better it will be for scientists to use their algorithms to investigate expansion prevention. It is also important to understand the impact of invasive species in general. Florida and Hawaii have the most severe invasive species problems in the United States. Florida has more introduced species than any other region of the USA and also ranks high globally. Exotic species will probably continually become introduced and established in Florida. This is not just a Burmese python in the Everglades problem. There are so many other invasive species in Florida, as well as other places that pose a threat to native fauna. Earlier, I mentioned how invasive snakes are becoming a problem worldwide. This research with ADLs can be used as a segue to help scientists all over the world deal with invasive snakes. Finally, I would also like to raise awareness to exotic pet trade. Many introductions of invasive species are due to exotic pet trade. I believe there needs to be more awareness of what people are actually getting into when they buy exotic pets. Because people were so obsessed with the idea of having a Burmese python, they didn't consider the needs of the python or the actual physical attributes of it either. As a result, many people release their pets into the wild. This problem not only affects the native wildlife, but also the introduced species as well. Because the Burmese pythons are not native to Florida, scientists and management officials are tasked with capturing and killing the pythons found in the Everglades. The pythons had no choice but to do what they needed to do to survive. It was not their fault that people were being irresponsible pet owners. So, if no other points today resonated with you, let these little points stay with you. So not only can you be aware, but also be informed of the problems that plague not just Florida, not just the US, but also the entire world. Thank you for watching my presentation.